Yeah, I remember Andy was saying, like, yeah, he was hard here because he just finished the capsule.
I got very excited. I saw that and I was like, good weather. Actually, it wasn't today.
Everything is
That's true. I'm actually not going to do anything. Just talking about like I just thought because of the weather. It's so nice out there.
Yeah.
Walking across the street, I know.
I came through the village, so I
No, I don't know. Okay. It's the sliders. Oh, good idea. After that, we're going to sign this. We had it for that 5 years.
5 or 6.
Oh, yeah.
Right. No, it's the 11 fingers of course.
I think she was just the moderator.
Yeah, it's like
I'm sure.
Okay, so we're going to get started on time today for you who are our regulars. I'm sorry to surprise you like this. And for those who are here for the first time, we do this every month. So welcome to the March edition of our monthly seminar on ethical, legal, and social issues in genetics. We are here in this room every month with people who are leaders in their fields from Columbia and from around the country. Let me just give you some coming attractions of the next couple of months, the rest of the academic year. In April, Dan Notterman is coming from Princeton. In May, Yoon Corey from the CDC and NCI, who's one of the leading thinkers and writers about public health uses of genomics. In June, Eric Turkheimer from the University of Virginia. Eric is one of the leading behavioral geneticists in the country and the world, and an interesting and skeptical thinker about knowledge in behavioral genetics. I think his talk will be fascinating. Today, however, we're delighted to have a homegrown crew to speak with us. Michio Hirano is professor of neurology here at Columbia. He will be joined by Dieter Egli, who is at the moment, I'm told, down on the main campus, but is on his way and will be here by 4:30 to do his piece. Dieter is with the New York Stem Cell Foundation and an assistant professor of pediatrics here at the Medical Center. And Mark Sauer, who many of you know, who's a professor of OBGYN here and heads the Division of Reproductive Endocrinology. And what they will all be speaking about is one of the most interesting and, as it has turned out, provocative new technologies on the reproductive genetics scene, and that is transfer of mitochondria to avoid the consequences of mitochondrial mutations. And the title of their talk is Preventing Transmission of Mitochondrial DNA Mutations via Mitochondrial Replacement Therapy. And Michio will get us going here. Thank you very much for joining us.
Okay, thank you. I don't see my co-speakers here, so we're in a little trouble here. But we'll start. Okay, so to start with my financial disclosures, I have some research support from a couple of pharmaceutical companies for work unrelated to what I'm going to talk about today. I'm a consultant for a small biotech company, and I'm the unpaid medical director of a diagnostic lab. So this is really a subject that's a merger of two very different fields. This is the mitochondrial disease field, which really started back in the 1960s with the discovery of mitochondrial DNA and the subsequent identification of diseases with mitochondrial biochemical defects back in the late 60s, early 70s. And then in the late 80s, the field really exploded with the identification of the first mitochondrial DNA mutations. And since then, we've been studying these mutations with Eric Scho, Billy DeMauro, and Bud Rowland, and others here at Columbia, as well as other people around the world. At the same time, in-vitro fertilization was evolving in the 1960s. It really started as a basic science. But then in the 1970s, the first child was born in the UK as a product of IVF. And subsequently, the first child in the US was born. And it's now quite mainstream. And Mark Sauer is going to talk to you more about IVF. Sorry, it's a little hard for me to see the screen here, but just to highlight, since, in the 2000s, these fields merged because mitochondrial replacement technology using IVF has emerged as a technique that is used for various purposes, but can be applied to prevent the transmission of mitochondrial DNA mutations, and I'll explain why in a moment. And it's even been used in rhesus monkeys to replace mitochondria. Here's Mark Sauer, and he'll be speaking in a few minutes. And this is actually started to be translated into human use to prevent transmission of mitochondrial DNA diseases. So let me give you some background about mitochondria. These are the powerhouses of the cell. They produce ATP through a process called oxidative phosphorylation that you learned way back maybe in high school. Certainly those of you who took science at college learned about it, probably learned those pathways and then tried to forget them. But the important points are that mitochondria actually evolved from bacteria, and they brought in with them the capacity to make ATP, but they also brought in their own DNA about 1 to 2 billion years ago. And they've retained, of course, that ability to make ATP. They form this symbiotic relationship with cells, and they retain also their own mitochondrial DNA, which is this small circular molecule. It's only 16,500 bases in length in comparison to the 3 billion bases of nuclear DNA. But it's essential. It encodes 37 genes that are required for these enzyme complexes 1, 3, 4, and 5, which are the ones that transfer electrons to generate a proton gradient, which is used to drive ATP synthesis. But the points are that here in pink, you can see the subunits, the protein polypeptides that are encoded by mitochondrial DNA. In addition, there are a number of genes that are required to translate that DNA into proteins. So obviously, mitochondrial DNA is essential to make energy and essential for life. This is a cartoon version of that mitochondrial genome, tightly packed with 16,500 bases of DNA. That the DNA has evolved over human evolution, starting with the so-called mitochondrial Eve here in Africa. And as humans migrated, that mitochondrial DNA has evolved into different haplotypes. And anthropologists have used these differences in mitochondrial DNA to trace the migration of humans. But it's so we all have different mitochondrial DNA. And one important factor is that mitochondrial DNA is strictly maternally inherited. Hence, we call the origin the mitochondrial Eve. That mitochondrial DNA is essential, as I mentioned, to make ATP. And so if you have defects of mitochondrial DNA, you can affect virtually every organ in the body. The brain and muscle are particularly affected, so these diseases are often described as encephalomyopathies. Well, in 1988, there was a major breakthrough, two major breakthroughs. The first mutations of mitochondrial DNA were identified. The first deletion, large-scale deletion, was identified by Doug Wallace's group in that same year. By Ian Holt's group in Queen Square, and then the first point mutation was identified by Doug Wallace's group in patients with a maternally inherited form of blindness. Since then, we've learned about over 270 different point mutations of mitochondrial DNA, so the field has exploded. We've learned that these mitochondrial DNA mutations are maternally inherited by and large. These mitochondrial DNA mutations can vary in amount. That's because there are hundreds or thousands of copies of mitochondrial DNA in each cell. So you can have variable levels of mutations from 0.1% to 99.9% mutation, and that is called heteroplasmy. And if you have high levels of these mutations, you're going to be more severely affected. And then it's important to know that because this is very critical in what we're going to talk about in trying to prevent these mitochondrial DNA mutations from being transmitted. These mutations also segregate differently in different cells in the body through mitosis. So sometimes cells will end up with very high levels of the mutation or low levels of the mutation and have mainly wild-type mitochondrial DNA, or in most cases a mixture heteroplasmic level of mutation. And of course, tissues that require lots of energy, like brain and muscle, are more vulnerable, and that's the threshold effect. Just to illustrate to you how one of these diseases, this is called MELAS. It was originally described here in Columbia by Drs. Pavlakis, DeVivo, Rowland, and DiMauro. This is a devastating, usually childhood onset disease characterized by atypical strokes because they affect young people, often children. These strokes lead to seizures and dementia and often early demise, unfortunately. These patients have elevated lactic acid because they can't produce energy through oxidative phosphorylation. They break down sugars into lactic acid, and they also have these funny-looking muscle fibers called ragged red fibers in muscle. Because of the heteroplasmy issue, the inheritance pattern is not always obvious when you look at a pedigree. Usually there's one person in the family, like this, who's severely affected with the disease, and in this case it was a child who had MELAS, who started out with weakness in about age 3, difficulty climbing stairs. She developed stroke-like episodes and cardiomyopathy and died at age 10. So it was a devastating disease. Her brother had some mental retardation. The mother, who was clinically unaffected, a maternal uncle, had mild symptoms of the mutation. He had some droopy eyelids and some ophthalmoparesis. So this is very strange. You see different disorders, or actually, in the case of the mother, no symptoms, and yet they all carry the same mutation. You can see that here. These are the measurements of the mitochondrial DNA mutation in muscle and blood of the various people in this family, and you can see that the maternal grandmother, who was unaffected clinically, carried levels of mutation that were rather low, 17 and 23% in blood and muscle, whereas the most severely affected child had very high levels in both blood and muscle. So this makes genetic counseling of these families very difficult, and it also makes it very risky for all of these women who carry the mutation to have children, because they may have a child with a very high level of mutation or low level. There's no way of accurately predicting on an individual basis. So it's really sort of a Russian roulette when it comes time for them to have children. So it's a very serious matter. And the diseases, as I pointed out, can be very devastating. One of the reasons why these mutations shift very rapidly from one generation to the next is it's a genetic, at least one genetic bottleneck, and there's probably a somatic bottleneck as well in development. So there's a genetic bottleneck during the early development. And so there's only a limited number of mitochondrial DNA segregating units that pass through and are transmitted to the eggs. So imagine if you have a jar full of, as Eric Schoen has taught me this analogy of black and white marbles, and you have thousands of black and white marbles, and you pick up, let's say, 20 of them. You could end up with 20 white marbles, which would be all normal, or you would end up with 20 black marbles, which would mean you have 100% mitochondrial DNA, or in many cases you'll have a mixture, and that heteroplasmy level can vary from 0 to 100%. So that's why these mutations can shift very rapidly from one generation to the next, and it's unpredictable how high the mutation load will be in the child. And of course, after the egg is fertilized, these cells divide, and so there's another bottleneck in mitotic replication of these cells that can also affect the level of mutations in different tissues. So these mutations are considered rare, but they're not very rare. In this epidemiological study in the northeast of England, they estimated that 1 in 10,000 people was symptomatic from a mitochondrial DNA mutation. And actually the number is probably a little higher. It's probably more like 1 in 3 to 5,000 was symptomatic from a mitochondrial DNA mutation. And in this study here from the same group in Newcastle, England, they looked at normal umbilical cord blood, well, presumably normal from healthy mothers and children, and they found that 1 in 200 people carries a mitochondrial DNA mutation. Fortunately, the levels of those mutations are low in most people, so they're never going to develop a disease. But that means that these mutations are out there, not infrequently. So this could be a significant problem. This was just published in the New England Journal about a week and a half ago. This was an estimate of how many women are at risk for transmitting mitochondrial DNA disease. So in the UK, about 2,000, almost 2,500, and in the US, over 12,000 women are at risk of transmitting these mitochondrial DNA mutations. And the number of estimated births per year among these women who carry these mitochondrial mutations in the UK is about 150, and in the US about 778. So significant number, not a huge public health issue, but it is significant. Just want to mention, this is Dieter Egli, he'll be speaking. We didn't have a chance to introduce you. Okay, so there is this technology, as I've alluded to before, to prevent the transmission of the mitochondrial DNA mutations. So Dieter will go over this in more detail, but there are basically two techniques. One is to do IVF and to take the pronuclei. So after the sperm fertilizes the egg, there are two pronuclei, one from the sperm, one from the egg, and then transfer those pronuclei to the cytoplasm of a healthy donor egg. So you do the IVF using the egg, let's say with a mitochondrial DNA mutation, and you transfer the nuclear DNA into the cytoplasm, which is mutation-free. And you can then implant the embryo and hopefully have a healthy child. And the alternative is to do the transfer of the nuclear material at the egg level, so I'm doing the mitochondrial or maternal spindle transfer, and then do IVF, and then implant the embryo, and again, hopefully have a child who's free of the mitochondrial DNA mutation. This has been done in monkeys, and this was a paper published in Nature about 6 years ago by a group in Oregon State. Shukrat Matalofov produced 4 baby monkeys after chromosomal spindle transfer of the oocytes and then in-vitro fertilization. And these monkeys apparently are quite healthy now. They're now about 8 years old, I believe. So they're mature monkeys now, and it shows that certainly this is technologically feasible. These monkeys, of course, didn't have mitochondrial DNA mutations. This has also been done in a mouse model. There's a mouse with a mitochondrial DNA deletion and in Japan. And what they did was they transferred the pronuclei from a zygote that had the deletion mutation into a cytoplasm of an egg, which was mitochondrial DNA mutation free. And you can see here that the males and females that did not have to undergo the pronuclear transfer lost weight and died early, whereas the ones who had the nuclear transfer lived or grew well and lived longer, and they did not develop the kidney disease that the mice with mitochondrial mutations develop. So that was the proof of principle, that this certainly can be done with a mitochondrial DNA mutation, but hasn't been done in humans. I just want to raise one concern that's been brought to attention recently. That's the possibility that in transferring the mitochondrial DNA or the nucleus into a different mitochondrial DNA background, you may end up with some interactions between the nuclear and mitochondrial genomes that may be deleterious. That's been shown in lower animals, in mice, in Drosophila, and other lower organisms, but it's never been shown in humans. And to our knowledge, here you can comment that there's no evidence that there's any mismatch between nuclear and mitochondrial genomes that would be deleterious. Is that a fair statement?
Carlos should then.
Well, that's between primates, between humans and monkeys, but among humans, there's no nuclear mitochondrial DNA incompatibility. Okay, so that's the background about mitochondrial DNA diseases. Mark, can I turn it over to you for the. I can put. Do you have the same slides webinar? Is that with your slides? Or do you have different ones? Okay.
Yeah, that's. You want to have a seat in front? It's where it changed.
Okay. Okay, just plug it in anywhere. This is today.
That's this one. It's right between my top. Just make sure it's talk. Probably is. Okay, sorry for the delay there. Was it 57th and Broadway at 3:45? It's a little out of breath. I'm lucky I made it as quick as I could. But anyway, thanks for inviting me. The three of us are kind of an interesting group, Dieter and Michio and myself. It's been referred to as an unholy alliance. Three Stooges, I heard that the other day. I don't know. Anyway, I want to try to give you a little background as to how, as a clinician who does research in reproductive medicine, got involved in this. Why, to me, it makes a lot of sense. And kind of where we are at with this therapy. I say a therapy because I think that's where it's going to be soon. And what's exciting about it. So with that in mind, my slides are coordinated because I've added things and subtracted things all day long here, so I hope I can give you the latest version of it. To start off, let me remind you that our field of medicine is rooted in kind of translational research, going back to Bob Edwards, who I had the pleasure of getting to know very well. PhD, a cell biologist who teamed up with a clinician who didn't know very much cell biology when he started, Patrick Stento, and with years of very secretive work in those days in order to achieve the first successful IVF, which didn't happen overnight, it happened over literally 2 decades, was successful. And today, most of the younger people in the room here just assume IVF has sort of always been a part of society. I can tell you it was not. When I learned it 30 years ago, it was a question as to whether it was actually worth learning because it was so crude and not very effective that we didn't know if it was worth our time to learn it as a reproductive medicine fellow. Clearly that turned out to be a good choice on my part. It's been controversial from the beginning. The Vatican tried to shut it down. The UK government in the early days tried to shut it down, as Bob Edwards would tell you himself if he were here. But today, 5 million babies have resulted. In my opinion, I would gather and guess that if Bob had to do it under the restraints of regulatory activities of today, we would have never been able to do IVF, or at least it would be very slow in coming into clinical practice. So it makes a lot of sense to me that we would get involved in this project, and it would be controversial, and that's really nothing new to me. I mean, most of my career we've struggled with whether it's egg donation or HIV treatment or menopausal mothers, all kinds of things I've been involved with. I'm not afraid of the controversy, but it seems to always focus on the more exciting projects. When I was asked by our dean last year to present to the Board of Trustees sort of a precision medicine overview, this is one of the slides I used, and I put it into this talk only because this idea of, quote, designer embryos, which is usually used more or less as a derogatory term, I would argue it's not. What we're really doing increasingly in IVF these days is providing better designed embryos, if you want to call it that, meaning better selected, higher success, higher normalcy than ever before, and lesser complications. So this project actually fits in very well with that kind of a mantra of well-designed embryos. The program we have, which I ran from this afternoon, is located at 57th Street. It's a big program. We do about 1,500 IVF cycles a year. We do a lot of clinical research. There's 10 doctors with me doing the work, full licensed laboratory. It's under the auspices of the FDA as well as New York State, which is a strict license. So there's a lot of regulatory oversight to regular in vitro, let alone the research aspects. I use this quote a lot because I just like it, but I think it's true for clinicians who do this kind of thing in general, which is once in a while, if you're lucky, and I've been lucky this way a few times in my career, you have the opportunity to do the right project with the right people at the right time and in the right place. And you really have to have all these factors working together to get things done and to move things along. And to me, when this association of PhDs that I work with and have worked with now for 5 or more years came about, I was delighted. And I'd like to think they were too, because in the history of this project, again, people were hitting walls to progress the science, to progress the clinical, and without some facilitation from various input from various people, this wouldn't have occurred. So what happened initially was I got a call from actually Robin Golan, again a clinician researcher, asking me if I thought it would be possible to do research egg donors here and pay them, because we do a lot of egg donation. And I said, yeah, of course, if you pay them. It seemed like an obvious question to me, but I didn't know where she was going with it. And about almost a year later, I was then approached by actually Kevin Egg and Scott Noggles about moving their work from the Harvard laboratories down to Columbia because we had access to egg donors because of the need to do stem cell research and do embryonic stem cell research so it could be directed and not contaminated and all these other problems they were having in Boston because you cannot pay egg donors to do research up there. It's against the law, as it is in California and other places. So essentially I said, yeah, I'd be up for that. I like those kind of challenges. And again, we were in a place, Columbia University, where we have a large pool of egg donors, so a ready pool of participants. And we had the willingness of multiple investigators to help them, and we had money. I have money to do these things. And we had good oversight. We already had at that point, obviously, an ethics committee, an IRB, and the stem cell committee was in place. And so we had all the basic things we needed to do this. And then in the background of all this, evolution is important. The times were changing, and we were seeing more and more national support, let alone the regional support and state support, to try to do this kind of stem cell research. So essentially what we did was, the first thing I was asked was, well, how do you know that the donors would be willing to be research subjects? And I said, well, I'll do a survey. So we surveyed our donors. And what we essentially found was that the great majority were perfectly fine being paid research donors, meaning that they weren't just there to produce eggs for reproductive purposes. They were basically there, as we sort of know, to be of service, but also to be compensated for their work, which in New York is an $8,000 per IVF cycle compensation. A very high rate, which made it even more controversial, especially when you're paying them to do research at $8,000. But we went to the ethics committee initially, and I posed the same question, and Ken Prager and the group agreed that it was ethical to continue to compensate egg donors at the same price that they are compensated to provide eggs for other reasons. There's nothing coercive, there was nothing enticing because this is what they already get. And we didn't solicit them any differently than we normally do, meaning that you wouldn't see things on the bulletin board saying, be an egg donor for research. It was, you're in our program, you've been screened to do egg donation. Would you also want to do this project? Yes, and the compensation is the same. And of course, well, I shouldn't say of course, because we didn't know it at the time. They were, in the great majority, happy to do that. In fact, what we have found over time is many of them prefer to do egg donation for research than for reproductive purposes, which is kind of an interesting project in itself. That led to a number of papers, and this isn't about the papers, but I just wanted to point out, I mean, in the last 5 years, there's been a wealth of very good basic science published in this diabetic stem cell research lab. That's where we started, as well as many spin-offs. And so, again, we're very transparent about what we do. The good, the bad, the ugly does get published. And as Dieter will tell you, it's not always been easy. Some of these projects didn't go as well. Some of them had to be redone. And again, these are expensive, very expensive projects, as you might guess, given that these are human eggs that are being used. So, getting more to the point of this talk, why do this project? Okay, and to me, it was an obvious reason because it's so compelling. And I won't repeat things because some of these things have been mentioned by Dr. Hirano before I stood up here. But these are awful diseases. I deal with women wanting to have children. I also deal with women increasingly with the PGD that we do who've had disabled children due to inheritable diseases. And these are really plagues on families, and they don't go away. They affect more than just the couple and more than just the child. They affect the entire social community around these families. There is, again, good estimates that these diseases that Michio told you about are in the community and they're not going away. And to me, one of the most unfair paradoxes to this is the couples coming in commonly didn't know they had anything wrong with them until they deliver a severely disabled child. And then the worst is the woman is typically told it's her fault because this disability came from her eggs. And therefore, if she wants to have more children and risk this, it's on her. Consider adoption, consider childless living. Consider egg donation so that you don't continue to basically contaminate your children. Or do PGD or prenatal screening, which we know because of the phenotypic variance that was described is not necessarily predictive of the outcome and therefore continues to provide risk. And last but not least, even if they are fortunate enough, as in this family, these two brothers, one affected severely, one not, shown in the corner down there, they will still potentially pass on disease in the females at least, as they express the disease downstream. And again, Michio mentioned this, I won't go into details to save some time, but there's been basically two methods. One where you switch the nuclei, well, you switch the fertilized egg, basically the nuclear material, from one to another. That's, I think, a harder controversy since this is an ethical discussion, I believe we're supposed to be having, because it destroys embryos. And that's always been a lightning rod for criticism with these methods. Or the therapy that we've been working on, which is just the maternal spindle transfers. And that involves eggs, but it doesn't involve destroying embryos. Is it ethically justifiable? Right. And these, as I could best discern from a variety of sources, tend to be the key ethical issues surrounding the clinical introduction of this. Are the animal experiments enough? And the counter to that, I'll kind of counter this simply, in my opinion again, as we go down the list. I'm not sure you can ever have enough animal experiments. I mean, where does it stop? At some point you have to be, I think, bold if you have enough information to at least believe that your benefits are going to outweigh your risk to move forward clinically. That's always a difficult decision. And I think when it's put to public debate, and I think when it's put even to peer review internally, it more people involved, the more difficult it gets, because you just will not get consensus. In the UK, I think, as I'll show you, has dealt with that pretty well, but you will never get consensus. We have enough animal data now, it's clearly safe. It's just not going to happen. Again, as mentioned, the potential mitonuclear genetic disconnects that can be caused, we don't know that that's a problem in primates or humans, but it's potential because of other animal studies, insect studies. The idea of modifying the germline, altering the genetics for future generations, creation of embryos for research purposes. Some people just don't like that idea at all. They don't like. There's a lot of people that, with all sincerity, that do not like at all what we do every day clinically. So they certainly don't like us creating embryos for this kind of purpose. And I'm appreciative of the social sensitivity to these issues. Believe me. The paying of egg donors, highly controversial. The National Academy of Science is not favorable to this. I mean, we're going against the grain, doing what we're doing, but we're doing it. Destroying embryos when performing transfers, as I mentioned before, in that one type. The unknown epigenetic and, again, downstream effects of the cellular manipulations. And then last but not least, the informed consent. One of the things that surprised me when we were down at Bethesda at the public meeting was the chorus actually of voices saying that the unborn or potentially disabled child that would result from an experiment that would go bad is not given adequate informed consent, and they need to be a party to these experiments. Now, I don't know how you consent someone that hasn't been born yet, but believe me, there are people that do believe that. It smacks of personhood for those involved in these kind of debates, which, as a member of the ASRM Ethics Committee, we're very concerned about that movement. It's not as far right and as hard to believe would ever happen as you might think. There are states that are close, if not already, passing these laws that would literally make embryos have the same legal status as you and me. And that would put people like me out of business, because every embryo we destroyed, it could be a manslaughter charge, right? So these are some of the issues that have been openly and very publicly discussed and certainly written about. You can find lots of papers on these topics if you're so interested. The thing I just wanted to spotlight, the Newfield Council Bioethics Group in the UK did a pretty exhaustive ethical review of this, and you can look this up online. It's a very interesting document. Essentially, their conclusion was that the science should move forward, that there was no justifiable reason to stop it based upon concerns over harm, that it needed to be obviously looked at carefully, that there was public support as well, but that it was an endorsement. And then the HFEA, Human Fertilization Embryology Authority, which is a governing group in the United Kingdom that has the power of law behind it, did, again, a 3-year analysis and continued analysis of the research being done in this field. They're the group that made the recent recommendation to Parliament to approve this for clinical introduction and further research. And again, the findings along the lines of what the Newfield Council had said, no obvious harm. The science was progressing well and encouraging, that there was broad public support for it. Obviously, also criticism. It's not a homogeneous support, but a majority support. And that there should be tight regulation under HFEA, which is, they already govern IVF in the UK that way. And if, in fact, in the course of the development of the technique, things were found to be harmful or obviously something that the public would not support, and be revisited constantly and potentially shut down. In parallel, but behind this, was what the FDA has been doing. The public hearing that was held that we went to last spring. I took that picture of Dieter up there at the podium. I don't think he knows that. I took that picture of playing his cell phone in the back of the auditorium. But it was a public forum. It was interesting because some of the best clinical and, more important to me, basic science researchers who have really done this work were there presenting it. And having watched it, presenting it extremely clearly, that 211, this is, as you can guess, is not easy to present to a lay public. because it was an open public meeting, which included a working group, which included our group, and included a group from Newcastle in the United Kingdom that had been doing this work and are spearheading this in the United Kingdom. And yet, the debate that occurred and that followed, I thought was troubling, very political, very lobby-stacked. One person after another on the microphone with what I would call pseudo-ethics credentials, arguing whether we were cloning people, and this, was Frankensteinian, the adjectives you commonly hear. But it was a good meeting in the sense that the work was put out there, and essentially they adopted a slow approach, go back to the bench, do more work, including work with human embryos, and that they would revisit it. And more recently, I've requested that the Institute of Medicine form a consensus report, which they're in the process now of doing, related again to this work, sort of along the lines of the New Field project in a sense. To me, they're sort of just replicating what the UK did, except going slow behind it. I think sadly, it's kind of, let's see what the English do, and then if they screw it up, we'll just, step away from it, as opposed to boldly going where no man has gone before, or whatever, as they say. So how do we proceed beyond the bench? Okay, so I know I don't want to cut Peter's time completely off here. Number one, we've been publishing this kind of work. Bob Klitzman and me have done a few papers on defending the ethics of this. Going back to 2009, the idea of paying egg donors and why we thought that was ethical. We published our opinion on that. The Nature of Medicine paper was a review of the mitochondrial replacement therapies and what they promised to do. Most recently, Bob and Klitzman and myself wrote a nice, I thought, again, kind of encapsulation of why MRT techniques and therapies are important to introduce and not to let the FDA just completely slow us down, including some op-ed rebuttals and Wall Street Journal op-eds. Again, the idea when we see things in the public that we think need to be addressed, we've stepped up and tried to address them. Because again, I learned from that FDA meeting, if you ignore the public, you're just being arrogant and they will drown you. So you've got to stand up when there's opportunities to address these things and defend it. Again, moving forward, it's important not to forget who we are. We've revisited this with the ethics committee, the specificity of this project of doing mitochondrial replacement therapy. Presently, we're submitted to the IRB. I knew it was close, and so we have an IRB project being reviewed. And although this doesn't involve directly stem cells, all of our work through the stem cell committee has been reviewed by the Dean's Committee on Stem Cell and approved. We never do these things in the closet. How would it be done? Briefly, I won't bore you with the details again. The idea is you take an egg donor, you enucleate her egg, and you put the nuclear material from the recipient, or the intended parent is probably the better way of saying it, the IP, and create the embryos. Now, in this case, for the project that's being proposed, we're not proposing to make babies, at least yet, but we're making embryos for characterization. And the value of this is not just finding the normal one, and that's going to be your baby. It's also all the embryos that will be produced, which is many more than the normal ones. We know this from normal IVF, make a lot more abnormal than normal. That material will also be available to study, also is going to give a tremendous amount of important information about this disease. It involves component parts of IVF. You have to stimulate women to get their eggs. You have to harvest them. You have to do the therapy I mentioned. To make the embryos by an IVF technology, grow them in vitro, ultimately biopsy and freeze those embryos for study. And again, these are things which we routinely do, so that's nothing unusual in our field of medicine to be able to do all these things we do every day. It does involve fertility medications. Those are injectables. It's not as bad as it sounds. I always hear these things, these powerful drugs that have to be, hormones that have to be injected. It's like, they're little quarter-inch needles. They're self-taught. It's not that difficult for people to learn how to do this if they're so motivated, obviously. I mean, if you don't need to take a shot, why would you? Monitored by ultrasound blood tests. We do this again routinely. It takes about 10 to 12 days to stimulate an ovary to produce multiple eggs or to make the multiple embryos. We monitor this. This is how ovaries look under ultrasound when you're doing this in the morning. These vaginal exams take all of 5 minutes. It's not too taxing. It's not painful. And they're done throughout the cycle. Like I said before, it takes about 10 to 12 days to get the group of eggs up. We take everything that you'll give, so to speak. These are 10-minute procedures. I'm not making that up. I was downtown today doing them. We did 5 today. And if you had a really efficient crew, which I don't, we'd probably do 5 in an hour. But you just kind of do them and clean the room and do another one. It's not that complicated. And this is done under anesthesia, propofol fentanyl, so you're sound asleep. But it's not surgery. You're not paralyzed. You're not intubated or anything like that. You wake right up. We watch it for an hour. You go home. That's IVF. Okay, that's how it works. This stuff is all, again, done in the laboratory. That's the beauty of, again, working in a translational science type field. I couldn't do this without my embryologist. They couldn't do this without a clinician, right? So it's a nice kind of a marriage. What we are basically doing is recreating what normally happens in the fallopian tube. So every day that we watch the embryo grow is shown on the bottom scheme there, all the way to the pre-implantation blastocyst is exactly what happens. And we all went through this in our mother's fallopian tube. Uterus gets all the credit, but it was actually the tube that deserves that. Is this expensive? And the answer is you bet it is. To do what I just showed you very quickly, 2 women in parallel with the medications, IVF on both basically. Nuts and bolts, probably $50,000 a cycle, a synchronized cycle. And then when you add your high taxes and the other things that go into this, you can imagine, right? So these are not going to be easy to do or cheap to do experiments, but I would tell you from my perspective and knowing the researchers that are involved, we could do this now if we were so bold. And I think we're in a good position to do it even before the English do it because we have the donors, we have the patients, we have the facilities, we have the investigators. So what's keeping us from doing this is not the money. It is the concern about stepping on the wrong people's toes, especially the FDA, and theoretically maybe shutting the whole thing down. So you always have to have, again, appreciation for the sensitivities of the people around you, and I think we've been very careful about that. On the same hand, the cost of not doing anything is huge. The babies that are born with these disabilities are severely disabled. Many of them certainly are spending $1 million in health care just per affected individual. So again, I would argue from a justice point of view, a resource point of view, and all kinds of other ethical points of view, it's the right, again, project to be doing at the right time in history. I always like this slide. Ruth Fishback over here has seen it before, but I always give a lot of credit to my friend Bob Edwards because this sums up why people like me get involved in this. It's all about having a baby between parents that really want this child. They want a healthy baby. They're expressing the love for the child and for their partner. That's what this is really fueling this. You can tell people, I see them every day, you shouldn't have children. Good luck with that. This is a profound effect on people. And I think we have the promise, at least, of helping an awful lot of people and change the course of reproductive history here. So I'll stop with that, and then I guess Dieter is coming up, right? Let me try to get this off, and I don't want to mess it up here.
Thank you. Well, I enjoyed the presentation. That was fantastic. So I'm going to pick it up from here and mostly talk about the practical aspects of how this is being done. How is the genome exchanged? How are these mitochondria exchanged between different eggs? mitochondrial disease. You've already heard about it, so I can skip that. But what I need to mention is that in the egg, the whole technique is essentially based on one principle, in that the oocyte genome, the nuclear genome, is located in a different place than the mitochondrial genome. What you see, I'm going to use my mouse. Here, the blue part is the nuclear genome, and it's attached to the microtubule spindle, and all those red dots, these are mitochondria. So the egg has a tremendous number of mitochondria, and several 100,000 copies of mitochondrial DNA. But the nuclear genome is essentially free of that. When you take out the nuclear genome, you take it out within an envelope of cytoplasm and surrounded by plasma membrane, so you don't actually expose it to a medium. You're not isolating any nuclear DNA here. It's always contained within a cell. And then when that genome is removed, that's what you've just seen here, it's removed within this little plasma membrane enclosed vesicle, and then it's transferred into another egg that no longer has its DNA. And what we did as a proof of principle first was to use a process called parthenogenesis. So what is parthenogenesis? It is development without sperm. When an egg develops, in order to make a human being, of course, you need a fertilized egg. But we reasoned that to do a preclinical experiment where we don't implant those cells, it's not yet needed to fertilize and make a developmentally competent embryo. Rather, we can make a so-called parthenode. And the parthenode, what it does, it simply gives the egg a stimulus, it starts dividing, and then in vitro it has the same developmental potential as a fertilized embryo. It makes a blastocyst, and you can derive stem cell lines from these. And these stem cell lines can then be looked at. Beta cells can be made from them, neurons and muscle cells, as I will later show. And then we can investigate the consequence of that transfer. Does it have an effect? How much mitochondrial DNA do we carry over? What's the effect on genomic integrity? What's the effect on gene expression? What's the effect on mitochondrial activity? These are all questions that we can address. And importantly, we will have no interference from the sperm genome. So that function of these cells will entirely rely on the interaction between the nuclear genome and those mitochondria where in that new egg. So this will allow us to address the question that came up earlier, is whether there is some sort of incompatibility between nuclear genome and the new mitochondria. So the first thing we looked at was simply development of those transferred eggs. Here you see the ongoing transfer. This is an enucleated egg, and here are two small vesicles containing the nuclear genome. You can't see the nuclear genome, but you've got to believe me, it's in here. And then, after transfer and activation, the egg forms a pronucleus, and that's now, here you can see the genome. It's inside this pronucleus. Then it cleaves, it's a 4-cell stage, and I always like to look at these pictures because human replication and development is just so beautiful. You have this 4-cell stage, you have here the 8-cell stage, then maybe 12 cells. In between there's the moral, and then you have these blastocysts. And some of these blastocysts have a good morphology with a distinct inner cell mass and this large expanding ectofectoderm here. And we were able to make stem cell lines from these. Dr. Sauer has mentioned that it takes retrieve 2 women, so you have to retrieve from 2 egg donors, and of course this has to be at the same time. And for that reason, we explored whether we can freeze the eggs of one donor, and therefore we'll have it easier to synchronize the 2 egg donations. And so it turns out that this is possible, and so this is a blastocyst derived after having one of the donors synchronized, cryopreserved the eggs. And what you see here is this is just a series of experiments that that look at what's the effect of the cryopreservation. So the higher the bar, the better the development, and the more blue is essentially the better the quality. So if you do just parthenogenesis without manipulation, you have about a 25% development. It's a little lower than if you do fertilization. So we take a little bit of a hit by doing parthenogenetic development rather than fertilization. But it's still efficient enough to do these experiments and address some fundamental questions. If you cryopreserve the egg, you slightly reduce developmental potential. But of course, we only have to cryopreserve one. So we can preserve the egg that is the nucleus donor. And you see here that if you cryopreserve the nucleus, the nuclear genome that is being transferred into a fresh enucleated egg, the development is actually very good. It's certainly as good as if you don't do the manipulation. So essentially, the manipulation in and of itself does not decrease developmental potential. It seems to be pretty benign whatever we do here to the egg. And that's, of course, very good to know. Also good to know is that this cryopreservation might make it considerably easier to synchronize the retrieval of eggs from two different women. Important is the question, how many mitochondria do we transfer? The answer is we do transfer some, but they eventually disappear. So what you see here, this is the number. So when you have mitochondria from two different women, there will be differences in the nucleic acid sequence of the mitochondria, and you can make PCR specific for one versus the other DNA. And you can quantify that. And so here, that's what we did. And so the higher the bar means the more mitochondrial DNA has been carried over from one egg to the other with that nuclear DNA. But you see here, it's very low. So this is in percentage. So it's less than it's about 0.3% on average shortly after is what we transfer. There's some variation to it, but it's certainly lower than 0.5%. So it's actually also close to the detection limit. And what we found here in these initial experiments is that when we derived stem cells, that this so-called heteroplasmy, that's how we call it, so the contribution from the carryover goes away, disappears. So these stem cells no longer carry those mitochondria. All the mitochondria are derived from the new egg. And you can see here that this is very stable. So you can culture these cells for 1 year and it's still the same. You can differentiate those cells and it still remains 0. You can do various things to those cells and that doesn't change. That exchange is stable, at least in these initial experiments. Here, so in these cases it was less than that many percent, so essentially it's actually completely gone. What you see here in this experiment is the quantification of the development. Again, this emphasizes parthenogenetic development. This is development after IVF, that's what I alluded to earlier. Development after fertilization is a little more efficient than parthenogenetic development, shown here, but it's not affected by the manipulation. Manipulation is about as efficient as no manipulation. We looked at the nuclear genome, whether that manipulation has any negative effect on the nuclear genome by looking at copy number variants. And the higher the bar here, the more copy number variants there are. These are unmanipulated cell lines, these are the manipulated cell lines. And you can see here it looks just the same. Some copy number variation in the genome is normal, but it's not increased by the manipulation. So that was good to know. We find that sometimes the chromosomes do not accurately segregate though. And that's something that is sometimes observed in human IVF development. It may be related to the method, but what is good to know is that we have methods to look at that and determine whether an particular embryo is characteristically normal. That's something that's very commonly done and almost routine, especially at advanced maternal age in IVF clinics. These are SNP arrays, and what you see here is that in this particular embryo, there was an increase, again, of this chromosome 4. So this is something we most likely will need to look at when such an embryo is made is, first of all, what's their mitochondrial genotype as a quality control? And second, how does their nuclear genotype look like? So a chief criticism that we have received